You want focus, but your day won't let you have it. You sit down to work, and your mind starts hopping. One tab, one message, one tiny, "I'll just check this," that turns into 20 minutes gone. That's the problem. Now, here's the plot twist. Some researchers say you can nudge your brain's timing with sound fast, on the scale of minutes, not years. The name you'll hear is 40 Hz gamma stimulation. Not a miracle, not mind control. Just a specific rhythm your brain can sometimes lock onto. I'm going to walk you through what that means in plain English, because you and I don't need hype. We need a clean map. First, think of your brain like a stadium full of people trying to do one job. If the crowd can't clap together, the noise stays messy. But if the claps line up, you get one strong beat. Brain rhythms work a little like that. Neurons don't just fire. They often fire in patterns. Those patterns show up as waves. You'll hear 3 wave names a lot. Alpha usually shows up when you're relaxed, especially when you're not taking in heavy information. You're awake, but you're not pushing hard. Beta tends to show up when you're doing active thinking, planning, talking, solving, scanning your environment. That everyday get it done state. Gamma is faster. And this is where the story gets interesting for you. Researchers often associate gamma activity with moments when the brain has to combine pieces, like pulling sight, sound, memory, and meaning into one understanding. Some scientists use the word binding for that. Separate signals becoming one organized picture. So why 40 Hz? 40 Hz is one specific speed within the broader gamma range. In multiple lab settings, scientists have tried using rhythmic sensory input, like pulsing light or patterned sound, to encourage the brain to synchronize to that rhythm. The careful term is entrainment. It doesn't mean you lose control. It means your brain activity can sometimes line up with an external beat, the same way you can start tapping your foot without thinking. And if that syncing happens, the hypothesis is simple. When different brain networks keep time together, information can move and combine more efficiently. That's the binding idea. Less scattered signal, more coordinated signal. But here's what matters for you right now. We're not chasing a vibe. We're chasing a measurable target, 40 cycles per second. And a measurable question. Does matching that rhythm make it easier for you to stay with one task? In the next part, I'm going to take you inside the mechanism, how timing signals can link distant brain regions, and why focus might be less about willpower and more about coordination. You want focus, but you also want to understand what's actually happening in your head when it shows up. So, I'm going to slow this down and make it practical, because you and I don't need mystery. We need a mechanism we can picture. Your brain is not one processor. You have many regions doing different jobs at the same time. One area handles what you see. Another helps you hold a goal in mind. Others track sounds, body signals, and memory. When you're focused, those areas don't just work harder. They work together on the same schedule. That schedule is what researchers mean by neural oscillations. It's a repeating pattern of activity. Think of it as timing. Not every neuron fires at once. Groups of neurons rise and fall in readiness, over and over. When the timing lines up across regions, signals can arrive when other neurons are ready to receive them. When the timing doesn't line up, the same signal can arrive at the wrong moment and get less traction. This is why more effort doesn't always fix focus. You can be trying hard, but if your brain networks are not coordinating well, you feel scattered. You start a sentence, then you forget what you meant. You open a file, then you check something else. Your attention doesn't stay locked because the system is not staying aligned. Now, we get to the core idea behind gamma rhythms and binding. Your experience feels unified. You see a screen, you understand words, you remember what you were doing, and you choose the next step. That unity is not automatic. Your brain has to connect features that start in different places. Researchers have long proposed that fast rhythms can help with that by providing a shared timing signal. If separate areas ride to the same rhythm, they can exchange information more efficiently. That's the basic claim. It's a proposal supported by a lot of observations. But it's not a single settled rule that explains everything. So where does 40 Hz come in? 40 Hz is one specific rate in the gamma range. In lab studies, when people or animals receive rhythmic stimulation at about 40 cycles per second, researchers often see activity in the brain that lines up with that rhythm. That is what entrainment means in this context. It's not a takeover. It's more like giving the system a steady beat that the brain can choose to follow, at least in part, for a period of time. If you're wondering why an external beat would matter, here's the practical angle. Your brain has to filter. It has to decide what is signal and what is noise. That filtering depends on timing. When you're focused, the networks that support your goal can become more coordinated, and distractions can lose influence. When you're not focused, the opposite happens. Competing signals win the timing battle. You don't feel one clear priority, you feel a pile of priorities. Researchers describe this coordination as synchronization or coherence between brain regions. The exact terms vary across studies, but the idea is the same. Timing alignment can support communication. If two regions rise and fall together, one region's output can arrive when the other region is most receptive. That can strengthen functional connections during the task you're doing. Now, I want you to keep one important detail in mind. Gamma is not a focus switch. Gamma activity can show up during many kinds of processing, and people don't all respond the same way to stimulation. Your state matters. Your stress level matters. Your sleep matters. Even the task matters. That's why responsible researchers talk about effects and probabilities, not guarantees. But if the mechanism is even partly right, it gives you a different way to think about focus. You don't have to treat focus like a moral virtue. You can treat it like a coordination problem. You can ask, what helps my brain networks line up and stay lined up while I work? That question sets up what comes next. Because once you understand the timing idea, you can evaluate evidence without getting tricked by big claims. In the next part, we're going to look at the studies people point to most often when they talk about 40 Hz, what those studies actually tested, and what they did not prove. You and I have the mechanism on the table. Now, you want the part that decides whether this is real science or just a clever story. So, let's talk about the evidence people point to most often when they talk about 40 Hz. In one of the most discussed early experiments, researchers worked with mouse models used to study Alzheimer's-related changes. They did something surprisingly simple. They delivered rhythmic sensory stimulation at 40 cycles per second. In that first wave of work, one method used flickering light at 40 Hz. The researchers reported two key observations. First, the brains of the animals showed activity that matched the 40 Hz rhythm, which is consistent with entrainment. Second, they observed reductions in certain Alzheimer's-related markers in the brain, including amyloid levels, compared with control conditions in that experiment. That doesn't mean a cure. It means that, in a controlled setting, a specific rhythm lined up with measurable biological changes. You should also know what makes this kind of result important and what makes it limited. It's important because it connects a clear input, 40 Hz stimulation, to clear outputs measured in tissue and brain activity. That's stronger than a vague claim like this feels calming. But it's limited because mice are not humans, and mouse models don't fully match the complexity of human Alzheimer's. In science, that gap matters. If you care about your own brain, you don't want wishful thinking. You want to know what the study actually tested. After the early work, researchers didn't stop at one sense. They explored whether combining senses could produce stronger or broader effects. That led to work using multiple types of rhythmic stimulation, like light and sound delivered around the same 40 Hz rate. The idea was straightforward. If different brain systems respond through different sensory pathways, then multisensory stimulation might engage wider networks. In follow-up research lines, scientists reported that multisensory 40 Hz stimulation could drive 40 Hz activity in additional brain regions and was associated with changes in measures linked to brain health in those animal models. The details vary by experiment, but the direction of the work stayed consistent. Measure the rhythm, measure the biology, compare to controls. If you're asking, how could a rhythm possibly connect to something as physical as protein buildup? Researchers have proposed several possible pathways, and you'll hear more than one. One proposed explanation focuses on how brain immune cells respond to changes in neural activity patterns. Another proposed explanation looks at a circulation of fluids around the brain and changes in how waste products may be cleared during certain activity states. These are active areas of research, and scientists debate which mechanisms matter most, or whether multiple mechanisms contribute. Now, we have to separate two questions you might be mixing together. One question is about Alzheimer's-related biology. The other is about focus. The Alzheimer's work often gets attention because it's high stakes and measurable. But that doesn't automatically prove a focus benefit in healthy people. It does, however, support a key point for you. 40 Hz stimulation can be studied in a rigorous way, and it can produce measurable brain responses under controlled conditions. So, what about humans? Researchers and clinicians have explored whether 40 Hz sensory stimulation is feasible and safe for people to use in structured settings. Some early studies and reports describe that participants can tolerate the stimulation and that researchers can measure brain responses consistent with entrainment. Some studies also report signals that might be beneficial in specific groups, but these are not final answers. Human studies often have small sample sizes early on, and results can differ depending on methods, duration, participant characteristics, and what outcomes are measured. That's the responsible way to hold this: promising signals, careful language, and an understanding that this field is still developing. If you're listening for a guarantee, you won't get one from honest science. What you can get is a clear picture of where the strongest evidence sits right now. Controlled lab work shows that 40 Hz stimulation can influence brain rhythms, and in animal models it has been associated with changes in Alzheimer's-related measures. In humans, researchers are testing feasibility and looking for reliable outcomes, but the story is still being written, and that puts you right where you need to be for the next step. Because once you understand what the studies do and don't prove, you can choose tools in a smart way. In the next part, I'm going to show you how people try to use sound-based methods at home, what binaural beats actually are, and how to use them safely and correctly if you decide to experiment. You and I have the science framed the right way now. We have a rhythm, a proposed mechanism, and evidence that stimulation around 40 Hz can drive measurable brain responses in lab settings. Now, you want the practical tool people actually try at home. That tool is often called binaural beats, and if you use it wrong, you waste your time. If you use it carefully, you get a clean experiment you can repeat. Here's the key rule you can't skip: you need stereo headphones. One ear gets one tone. The other ear gets a slightly different tone. Your brain can perceive the difference between them as a third rhythmic beat. That perceived beat is the target. If you want a 40 Hz beat, the two tones need to be 40 Hz apart. For example, 440 Hz in one ear and 480 Hz in the other ear creates a 40 Hz difference. You don't need those exact numbers, but you do need the difference. Now, set this up like a real test, not like a vibe. You pick one task that matters to you, one writing session, one study block, one planning sprint. You don't multitask, you don't scroll. You decide what focus means before you start. For you, it might mean I wrote 300 words, or I finished 20 practice questions, or I cleared my inbox for 15 minutes. You measure something simple. Keep the volume comfortable. If it feels sharp or irritating, it's too loud. Louder does not mean better. The goal is steady exposure, not intensity. You also want a clean audio track without extra effects that distract you. Some people add soft background noise under the tones. That can be fine if it helps you tolerate the sound, but don't turn it into a playlist you start analyzing. You want boring. Boring is good. Now, timing. If you're trying the 15 minutes idea, treat it as a warm-up. You listen for 10 to 15 minutes, then you start your work immediately. Or you listen while you work if the sound doesn't pull your attention away. Either approach can be tested. What matters is consistency. You keep the same start time, the same task type, and the same duration for a few sessions so you can compare. You also need to know what this is not. Binaural beats are not a guarantee. They don't force your brain into anything. People respond differently. Your sleep, stress, caffeine, and expectations can all change what you feel. And feeling focused is not the same as doing focused work. That's why you measure output. If you're sensitive to repetitive sensory input, start gently. Short sessions. Lower volume. And if you notice discomfort, stop and reassess. You're not proving toughness here. You're testing a tool. One more practical point. Speakers are not the same as headphones for binaural beats. With speakers, the tones mix in the air, and your ears don't get the clean separation needed for the effect. Headphones give you that separation. So this is your experiment. Stereo headphones. 2 tones with a 40 Hz difference. Comfortable volume. A single task. A simple metric. And no distractions. You're not chasing a promise, you're running a repeatable test on your own attention.